Nice to see some familiar faces here and many people who I've not met before. What I thought I wanted to do today is to really just exemplify the fact that we're not just sitting on our laurels. The international community that are interested in this disease continue to do research, trying to gain a better understanding of what makes GISTs occur, new developments in terms of trials, and new drugs to treat the things that we haven't cracked yet. While it's true to say that the majority of patients have a particular type of GIST which we can effectively treat with imatinib, that ignores the fact that for many of those patients it's only a temporary respite of a few years, many more years than it used to be because of new drugs, but it's not a cure. And there are still some patients where the existing drugs don't work. So let's just focus on that to start with. These are the real problems we've got at the moment. There are some types of GIST which don't respond to the current drugs. Don't worry about the numbers and so on at the moment. I'm going to explain that in a second. We've got some patients where the drugs work for a while and then the tumour evolves to get round the problem. I mean, to get round their problem, which is the drugs are working. They evolve with resistant mutations. They change, and then the drugs stop working. And first of all, we can introduce sunitinib after imatinib. That may work for about half the patients where imatinib has stopped working. We can try regorafenib, and that works for another percentage of patients, but only for a while. And so we need constantly to be developing new drugs which can overcome this, and probably even more important, develop strategies which are going to delay or prevent those resistant changes taking place at all. And then we would really be able to say that we've turned GIST into a chronic disease, which is, I think, at the moment, the best we can hope for. Cure, of course, is the ultimate goal. And then, of course, we have the problem, as Jane has said, of the wild type and syndromic GISTs, which are difficult to treat. We have some treatments, but for some of those, we don't have good treatments at all. So just for the benefit of people who are unfamiliar with this, most GISTs are driven by a change, a mutation in a gene. That's a bit of DNA that tells the cell how to make a protein. And that means that the protein is different. And in this particular case, it means that it is switched on all the time, constantly telling those cells to divide and grow and spread. In the majority of cases, it's in this protein called KIT, and the KIT gene, and it doesn't matter particularly where, but it's exon 11, and many of you will be familiar with that because many of you who are patients will have a tumour with one of those mutations, and usually they respond to imatinib. And for some patients, we don't know exactly. That's partly why we want to do some more research. But for some patients, probably something around 15% of patients, the resistant mutations never occur. When they do occur, they occur in other regions of the gene, and sometimes we can do something about it, sometimes we can't. Now, in about 7.5% of patients, the change is in a different gene, platelet-derived growth factor receptor alpha, PTGFR8, And usually it's in this bit called exon 18. Now, unfortunately, when it's there, the standard drugs don't work. And it's a real challenge at the moment to find any drugs that do work. So let's look at that first of all. So this is the so-called D842V. The D and the V stand for their shorthand for different amino acids. And those changes make the gene create a protein which has a different building block in it, and that changes its function. So this is a drug, crenolinib, and there was a phase 2 study reported last year at the American Society of Clinical Oncology meeting. 12 patients with the precise mutation and progression, and looking at PET scans, positive emission tomography scans, 3 patients had stable disease, didn't look worse. But if you looked at the tumour size, one of those was stable, one of those grew, one of those shrank. And then one patient had a tumour which went completely dead looking on the scan, on the PET scan, but it actually grew. So that's a bit confusing. And one of the one that they reported where it looked a lot better, there it is, great big hot lump down in the pelvis, and the uptake of the radioactive glucose switched off within 3 weeks. Now it's probable that the growth was spurious and that patient was genuinely benefiting from treatment. But as you can gather, it's not really clear how good a drug it is or where we're going with that. More trials need to be done. So there's another drug, and this is a more recently developed drug called Blue 285, developed by a company, and it's an academic sort of venture capital funded small biotech company called Blueprint. So Blue 285 was developed specifically for some of the mutations we can't treat at the moment. And that includes this difficult one, the exon 18 D842V, and also some of the mutations that develop in patients on imatinib or sunitinib that we can't currently treat adequately. I won't go into the technical bit, but it's in a different part of the protein from the true business end, where imatinib, for instance, binds and stops the energy molecule ATP getting into the heart of the enzyme. And it's in another bit that's sort of regulatory. So that's one of those resistant mutations. So it develops in patients who are on standard therapy. It's not there to start with usually. It develops as the tumour evolves to overcome the treatment. And at the moment we can't treat it very well. So this compound is in early clinical trial. A number of these resistant mutations have been looked at and appear to be sensitive to this new drug. It's more selective than, it's more targeted, if you like, than crenolinib. This is it's like an evolutionary tree, this, but it's actually telling you which proteins a drug is capable of hitting, and crenolinib is what we call a dirty drug. Sometimes we want that. It inhibits a host of different proteins, whereas Blueprint 285 inhibits just a few, but it's the ones that we want to inhibit. Now these numbers up here don't mean a lot to you, but the number basically is telling us how effective the drug is, and the smaller the number, the better. So these are types of GIST that imatinib doesn't work in, and as you can see, these are big numbers. Crenolanib looks pretty good for this one, D842V. Not too bad for this one, but it does a lot of stuff that we probably don't want it to do, and some of that may not be helpful. So at least in the laboratory, this drug is looking good. So there's a trial. I won't go into this in detail. This just tells you which sorts of patients are going into the trial and what sort of laboratory tests are being done to try to work out whether it's working or not. I've got no information on the trial because it's too new, but I think it's promising that people are developing new drugs specifically focused on those types of GIST which are either resistant to treatment from the start or develop resistance to treatment that currently we don't have very good drugs for. There are other drugs. So Ponatinib has some activity in this area, particularly these evolving resistant mutations. It has a rather checkered history as a drug because it was tested at a dose which seemed to be too high and was causing a lot of problems with blood clots and blood clots coming off veins and ending up in the lungs, or blood clots in arteries in the brain, quite toxic. So they've had to stop the trials, redesign the trials with a lower dose, start again. And this is not in GIST, this is other diseases. And at the moment it's not clear whether there is a plan to develop Ponatinib in GIST. We thought we were going ahead with a trial about 18 months ago, and then it was on hold and hasn't been restarted. And another drug called cabozantinib, which inhibits quite a number of different proteins, including relevant ones, and there are plans to test that in patients where the standard drugs have failed. We did a trial with a combination of imatinib and a new drug that inhibits a different protein entirely called PI3 kinase. It just had a code name BKM120. It was a drug with Novartis. We saw some evidence of activity. There were some unusual side effects that could be difficult to manage in patients who are having long-term treatment. The study hasn't been published yet, and we don't quite know where this or a different trial with another PI3 kinase inhibitor is going to go. It would require a big trial, and you would have to have one group of patients treated in a standard way and another group treated with the new combination to really work out what was going on. Another approach, which could be promising, is to use the standard drugs we've got already, but use them in a different way. And a trial called Alta Geist has been very long in the gestation. I first discussed this with colleagues, I'm sure it's 3 years ago, if not more. It must be more than that. The idea came from a group in Australia, and excuse me, a guy called John Salzberg in Australia has been involved in GIST research right from the very beginning, and he figured that if you could alternate between imatinib and another drug, regorafenib, has been chosen, you might stop these resistant mutations developing. And so the trial is designed as one group have standard. This is patients who need a drug for the first time, so advanced metastatic, either imatinib or imatinib for 3 weeks, stop the drug for a few days, regorafenib for 3 weeks, stop the drug for a week, and keep switching from one to the other. Now, the trial is accruing quite slowly, and there are concerns about that. I think it's difficult because when we started doing research with imatinib in the year 2000, 16 years ago, more than 16 years ago, I treated my first patient in September 2000. We were seeing all the patients who were available in academic centres, sarcoma centres, and therefore all the patients needing treatment for the first time would come to a place where clinical trials were being done. Nowadays, it is so easy to prescribe imatinib, and everyone who treats gut cancer can do so, and many patients are on treatment with imatinib before there's ever any involvement of a centre that's doing clinical trials. And that's a problem, because then it's difficult to introduce the idea of doing something experimental, especially when the patient may already know within a very few weeks that the imatinib is working. So there are big challenges to doing this sort of research. Wild type, it's a misnomer. We didn't know that when we called it that, but it's supposed to, well, what it does mean is that there are no mutations in those two genes I've talked about so far, KIT and platelet-derived growth factor receptor alpha. But there are situations where other mutations can be the problem, and the mechanisms are different. So in the majority of cases of wild-type GIST, we'll call it that, but to use the acronym for the clinic, PAWS GIST, that's the pediatric, adolescent, wild-type and syndromic. So just to encompass all of those, and they are different. In the majority of cases, an enzyme that is crucial, and I'll try my best to explain that in a moment, called succinate dehydrogenase, it's lost. That crucial function of the cell in metabolizing energy, glucose, is lost. And that has big repercussions. So sometimes that loss is inherited. So one or other parent has lost that enzyme in the gene. So the gene for that is permanently damaged, and that is passed on to children, and that's called Carney-Stratakis syndrome. It's also associated with another type of cancer called paraganglioma. In more cases, many more, I know it's very rare, but many more, it's not inherited, but there is a mutation. So it's just happened, arisen spontaneously in the tumour. So there's a mutation in there are 4 genes, STH, A, B, C, or D. We call that STH-X for shorthand. Now that can be in children or adults. Sometimes it's lost and we don't yet know exactly how. We do know what happens, but we don't know the gene. And usually those are the cases that arise in young teenagers, usually female, not always, They're usually gastric. By the time they are diagnosed, they're often in many different places. And as I've said, they're usually female patients. And then we have this other syndrome called Carney triad, where there are 3 genes: GIST, paraganglioma, and curious tumours in the lungs called chondromas. And they look like great big lumps of calcium on a chest X-ray. Those are very rare patients. And at the moment, we don't know what the gene is, curiously. Now, there are 2 other groups. Neurofibromatosis, used to be called von Recklinghausen's syndrome. It's the problem that afflicted the very famous patient called Elephant Man. Some of you may have seen the film. It's a syndrome where you have multiple benign lumps which can occur in the skin and in nerves throughout the body, and sometimes a change occurs which is cancerous, and sometimes that creates a GIST. And then you can have a mutation in a gene called BRAF. That occurs much more commonly in melanoma, the pigmented skin cancer that is so aggressive. In those cases, there is a drug, vemurafenib, and other drugs which can treat it. At the moment, we don't have, I think that's the next slide, we don't have a drug for neurofibromatosis. Now, I want to introduce a very difficult concept. DNA is composed of, building blocks which create the genes that tell the cell how to make protein. But the chromosomes, where the DNA is stored, are not just DNA, they're DNA and proteins. Those proteins are called histones. And because the DNA is so tightly packaged, it has to be unraveled in a special way so that the genetic code can be read. Otherwise, it's just too tightly triple wrapped and scrunched up, and you can't get at it. And that process of unraveling is actually very tightly controlled. And one of those bits of control is to put methyl groups, that's a carbon and 3 hydrogens, onto these proteins that changes their function. That's called methylation. And one curious thing is that there's a group of succinate dehydrogenase deficient wild type where there's no mutation at all, and that is always associated with methylation, specifically of SDH, succinate dehydrogenase gene C. At the moment, we don't know why. This is work of Paul Meltzer and colleagues at the NCI in America. Now, I'm going to try and introduce explain a little bit of what I've just said. That's an inner city ring road. That's Sheffield. For those who live in the south, you'd be more amused by saying Basingstoke, because it's sort of famous. And what do you think about when you see one of those? You think, oh no, if anything happens, I'm going to be stuck here for hours. The M25, of course, is also been described as one of the biggest car parks in the world. So if something happens on a roundabout, on a ring road, the next thing that happens is that. I like the fact that people can get out and walk their dog, I mean, that's probably quite useful. But if you're trying to get somewhere, it's not much fun. There's a backup That's what happens. Now, the Krebs cycle, which is where succinate dehydrogenase fits in, is not a very popular topic with medical students. And I was sent that by one of my junior colleagues. I thought it was very amusing. It says cycles attached to these railings will be removed, and a picture of the Krebs cycle, just waiting for someone to rip it away. That's what it looks like. You don't have to you don't have to understand this. I wish I did. But we're looking at one particular enzyme that changes this molecule into that molecule. Now, what happens if there's a block here is that succinate, being this side of the block, builds up, just like a traffic jam. And that has tremendous repercussions. If you have too much of one particular compound, and you've got a process where that is involved in changing one thing to another, if you've got too much of one of those, that process stops. And one of those processes is the degradation of a protein called HIF, which is hypoxia-inducible factor 1 alpha. That responds to oxygen in the body, and if there's plenty of oxygen, it is constantly degraded, so it doesn't build up. If there's a lack of oxygen, it does build up, and then changes occur in the body that try to adapt to the lack of oxygen. Now, when there's too much succinate, the body adapts as if there is not enough oxygen. This is occurring in the tumour, remember, which means that some of the proteins that wouldn't normally be there accumulate. And one of those is called vascular endothelial growth factor, VEGF, and VEGF drives cancer. It drives blood vessels to be formed. It inhibits the immune system by changing the function of certain lymphocytes into a state where instead of helping kill cancers, they actually help cancers to evade the immune system and grow, and a host of other things. And also, the same process, succinate buildup, changes the methylation, this change on the proteins in the chromosome, in a bad way. And so, in a nutshell, we've got mutations, or which can be associated with wild-type GIST and paraganglioma, and we've got changes in DNA methylation, which can alter the function of many genes. They can be switched on or switched off, but they're usually switched off. And this is characteristic of wild-type GIST. So to put that into a nutshell in terms of treatment, we've got an increase in that protein VEGF, and drugs that inhibit the receptor for that protein where it works, which is VEGF receptor, include two of our drugs, sunitinib and regorafenib. And that's why sunitinib and regorafenib are useful in treating wild-type GIST. Currently, for the NF1-related wild-type GIST, we don't have any specific treatment. There are some ideas about combining some of the new drugs that are still in development. They're not sort of on the shelf yet, but that might work, but we're not there yet. And as I said, for BRAF mutant wild-type GIST, which is ultra rare, we do have some drugs like vemurafenib. So clinical trials, there is activity at the moment. There's a regorafenib trial for first-line treatment going on, I think mainly in the States. It's recruiting currently. There's a trial with vandetanib, which inhibits several of the important proteins, which has recruited a certain number of patients. They're waiting, doesn't say on the trial website what they're waiting for. They may have all the patients they need, or they're just waiting to see what has happened with the patients that they've treated so far. So that's still active but not currently recruiting. And there's been a trial with lincitinib, which inhibits another protein called insulin-like growth factor 1 receptor and insulin receptor, which are both upregulated, both increased in the tumour cells in wild-type GIST. That trial's completed, but as yet we haven't heard any results. There was a previous trial with an antibody against this protein, which didn't show as much promise as we'd hoped. Let's just put it that way. I don't know. And there's a possible role for drugs that inhibit this process of putting methyl groups onto DNA. That is speculative, and at the moment we're waiting for some newer drugs other than the ones that we know about, like azacitidine and decitabine, to come along. And there are some drugs being developed, and that's what one of them looks like. So I like pretty pictures. So my conclusions really are there's a lot of clinical trial activity in GIST. Some novel targets are being tested, like PI3 kinase. There are trials with drug combinations. There are trials with alternating existing drugs. And our understanding of pediatric adolescent wild-type syndromic pause GIST is improving. We're learning more. But at the moment, we can't say there are any recent breakthroughs. But I think it gives one hope that things are continually going to improve. I'll stop there and happily take questions. And if you want to go back over some of those pretty pictures, by all means. And I apologise for the complexity, but I wanted to just at least give you a flavour of what's going on.
I've got a question. Can I borrow the microphone a minute?
It's turned off.
Okay.
It's turned off.
Okay. Yeah, my question relates to the ALT trial. If they're struggling to recruit people, why can't they take patients who are already on imatinib?
They can. They can be on imatinib for a month before they go into the trial.
Yeah.
So they haven't. But not if they've been on for 3 months, 6 months.
But why is that?
I think that I think it's probably just not regarded as a good test of the treatment. I didn't design the trial.
No.
It was designed in Australia, and I knew that we would struggle at the Marsden to recruit patients because we see relatively few new patients who have not had prior treatment. We do see some, and particularly those who come to us through our own GI unit. But it's a problem. I think that if patients aren't informed about clinical trials, and it's difficult to inform them about clinical trials at a stage where they're only just learning about their disease. They haven't had time to get in touch with groups like this, and where they're in a centre where no clinical trials are taking place. So it's a challenge. I'm sure it can be overcome, but it is a problem.
Does anybody else have any other questions on that simple
One here, one there.
presentation we just had?
Sorry.
Yes, you mentioned earlier in the talk about drugs like Panetta. In the UK, it's not clear to me if there's any possibility of access to these sorts of drugs which aren't on the NICE list or the CDF list.
I think that drugs that aren't licensed, in other words, they aren't on the market for a particular cancer, it's extraordinarily difficult, I would say virtually impossible, to get hold of those drugs outside of a clinical trial that's running. Mainly because, I mean, Imatinib particularly, it's toxic. It's not as straightforward a drug to give as Sunitinib, Regorafenib, and there are concerns about not knowing what's the right dose, not knowing if it's safe. So a company, it's a tiny company anyway, they're not going to release it just to individual patients because they haven't got the proof that it's safe to do so.
Professor, thank you. How far away are we from being able to predict resistance at an individual level rather than at a statistical level?
Well, the way that we think that that might occur in the future is by looking at DNA in the blood. I won't say, I think predict is probably the wrong word to use, because if the resistant mutation, we can predict resistance if it's the D842V. In fact, so much so that when we consider patients for adjuvant treatment, if they've got that mutation, we don't give it, because we know that adjuvant imatinib would not work. So it's saving the patient toxicity, it's saving the nation money. And so that's prediction. But if the resistant mutation that arises in patients on imatinib only arises because of the selection, the driving force of the treatment, which is telling the tumour to change, to get round this inhibition. That's how the evolution works. That mutation is probably not there to start with. So one cannot, at the moment, predict. What we could do, and the technology is evolving to allow this, and is being incorporated into trials like the BlueGist trial, is to look at the DNA that's in the blood, where you can now, technically possible, identify a resistant mutation in the blood, and that there will be a need for a new treatment at some point, and perhaps those patients might be selected to go into a trial of one of these novel alternating schedules, for example. Because the difficulty of biopsying, actually taking a sample from a tumour, is that different mutations can occur in the same tumour or in different secondary tumours. And as we often see, we often see a patient where one liver metastasis grows, the others don't. And we can treat that one that's growing either by cutting it out or by burning it in some way, radiofrequency or another sort of ablation technique, and then keep the patient on standard therapy. Sometimes that works for years. I've got one patient where we did that 11 years ago, and he's still on imatinib. That's exceptional, but it does highlight the value of that. But prediction would only work if those resistant mutations were there from day one, and they're probably not. And even if they were, it's one cell in 100 million, and we're not going to detect that DNA.
The data that you've collected with your 550 patients, is the data from that likely to give us any clues as to the predictive behaviour of current patients?
I think it might do, and it's going to require some more in-depth analysis. But what we're already learning in collaboration with other teams who are doing the similar work is that there are certain types of mutation, even in exon 11, which are less, which have a better outlook than others. So where a big chunk of the DNA is lost, is missing, as opposed to just a switch which changes one building block, one amino acid. If a big chunk is missing, that seems to be associated with a better outlook. We don't know why. And there are, on the other hand, other mutations in a sort of slightly different part of the molecule which have a worse outlook.
So exon 11s are not all the same?
Exon 11s are not all the same. Yeah.
A slightly different question.
Yeah.
How important is it to you that your patient is aware of exon 11 or exon 18 or that information or any mutation?
I think it's the sort of information which we're very happy to share with patients if they ask. It's is it important that they know and understand it? I think we are sensitive to the needs of individual patients in terms of their understanding of the disease and how much they want to know. Because we believe it's important to have that information from day one, we routinely test. So when we diagnose a new patient with GIST for the first time, we always do that analysis. And generally speaking, we would share that information with patients. But how important is it for them individually to know? I think it helps if we can give them some broad idea of how long treatment is going to work for. So, we know that a different mutation in exon 9 is associated with a shorter period of disease control compared with exon 11. And sometimes a bigger dose or a different drug might be better for that patient. And we would certainly, in a patient with an unusual mutation, we would certainly share that information with the patient because it's going to change the way we follow them up. It's going to change the way we treat them.
Right, okay.
Anybody else? Okay, time is running short,
sure.
so thank you, Ian.
Okay.
Although that was complicated, I think that those of us that have been to a number of meetings tend to find that sort of complicated information becomes a little bit more familiar. So the next meeting that you come to, it'll make a bit more sense to you.